How's it going, guys? Past level question for microbiology for step 1 researcher. Conducting an experiment, antibiotics resistance genes. Bacterial strain A is resistant to vancomycin, bacterial strain B is not. You grow both bacterial strains together and then holy shit, bacterial strain B is now also resistant to vancomycin. Which of the following supports conjugation as the mechanism of antibiotics resistance gene acquisition? Let's just hop through. Choice A, direct cell-cell contact, correct answer. Simple definitions here. So conjugation is when you have a tube called a pilus, which is going to connect to bacterial cells. You have an F plus cell, which has a plasmid, which is a circular DNA arrangement, which is almost always where the antibiotics resistance genes are located. Sometimes they can be located in the bacterial chromosome, but usually they're on the plasmid. And you're going to have this tubular structure, the pilus, via which the plasmid is going to move from the F plus bacterium to the F minus bacterium, the latter which does not initially have the plasmid. So if you know conjugation involves two bacterial cells coming into direct contact via a pilus protein, that answer obviously makes sense here. And if you think it's a little bit weird or you're not sure, you can obviously eliminate to get there, which is also what we'll do now. So let's just hop through the others. Choice B, disruption with the addition of nucleotidase. Wrong fucking answer. And we know it's wrong because this is transformation, which is when you have a bacterial cell acquire/grab DNA fragments directly from the extracellular matrix/agar. So strep pneumo is a classic transformer where they'll tell you researcher conducting an experiment, there's observation of disruption of the antibiotics resistance gene acquisition when DNase is added to the medium, which the following is the most likely mechanism of engagement between the bacterial cells, and the answer is transformation. Okay. Whereas with conjugation, for instance, the tube is protecting the genes from being degraded by the addition of DNase. Okay. Wrong fucking answer. Choice C, lysogenic phase. Wrong fucking answer. Refers to specialized transduction. Transduction means you have a viral particle, aka a phage particle, that invades a bacterial cell. And if you have the viral DNA incorporate itself into the bacterial DNA, we call that lysogeny, lysogenic process. It's a vocabulary word you need to know. Now, it often occurs in nature under unfavorable environmental circumstances, aberrant temperature, acidity, hydration, etc. So viral particle can go latent within the bacterial DNA, and then upon favorable environmental circumstances, we can get reemergence of the viral DNA from the bacterial DNA, where it will flank adjacent sequences and incorporate the totality of that nucleic acid, the viral plus the flanked bacterial sequences, into new phage particles, which then leave the cell and invade other cells, and that's how we can get antibiotics resistance genes transferred. Now, I said they're usually located on the plasmid, so the phage particles can incorporate plasmids in addition to other sequences, but it's the mechanism of the lysogenic process that the phage particle engages in, which is specialized transduction. Choice C, wrong fucking answer. Choice D, lytic phase without lysogeny. Wrong fucking answer. Refers to generalized transduction. So you have a phage/viral particle invade a bacterial cell, and then without there being any type of incorporation into the bacterial DNA, you're simply going to have new phage particles synthesized where plasmids and free DNA floating around the bacterial cell can be packaged into those new phage particles where they in turn invade other bacterial cells, and that's how we have transfer of antibiotics resistance genes. Wrong fucking answer. Choice C, viral DNA flanked by bacterial DNA. Wrong fucking answer. And we know it's wrong because, as I just harped on, this is specialized transduction. It's the same thing. So if you knew that C and E are the same, then you could eliminate them in this case. Wrong fucking answer. You know the deal. To make more content, if you like my stuff, subscribe my channel. Appreciate your time. That's it.